Now we are going to discuss about our next topic, which is the chromosomal theory of inheritance. We need to understand that who are the scientists who contributed to Mendel's work, because when Mendel's presented his law, there was no one to represent that how this can be a law, because at that moment chromosomes were not discovered. So there were three European scientists who started working on this. So these scientists were Carl Corin, Hugo von Varis, and we have another name as well, Carl Corin, Hugo von Varis. Von Varis, right? It's not von Varis, it is von Varis. Hugo von Varis and von Steruska. I'm not sure about the actual spell, you can just look into it. Carl Corin, Hugo von Varis, and von Steruska. Steruska. It's M actually, Steruska. So these were the three scientists who recognized the work of um, you know recognized the work which was being done by uh, you know George Mendel, and started working on it. These all of them were European scientists. After which, when Walter Fleming discovered the chromosome, he there were certain scientists who worked on actually hypothesizing that what can be the role of chromosome in heredity. So at that time, it was not known that chromosome played a central role in heredity. But the first scientist who came up with the idea, who observed actually and gave the idea of that chromosome plays a role in heredity, is Carl Corin. Carl Corin, he just republished. He published his observation. What he did was he was uh, what he did was he actually published this idea that chromosome is a hereditary material in you know in a paper with reciting the work of Mendel. The paper's name that was named Mendel's. The paper's name was Mendel's trait concerning the origin Mendel's trait concerning the origin of chromosomes chromosomes of racial heredity. This was the name of the paper which he published at that time, and soon after the idea which he had observed, another scientist named as Walter Sutton. Walter Sutton was basically the student of he was a student at California University. He was a graduate student of E.B. Wilson, and what he did was that he actually first time formulated the chromosomal theory of inheritance. He was actually observing the rapidly dividing sperm and egg cells of a grasshopper, and at that moment he just found out that the number of chromosomes which are present in the number of chromosomes in the egg, which are present in the egg and present in the sperm, were equally contributing to the traits which were present in the offspring. So at that moment, what he actually observed was that there was an equal contribution of chromosomes from the egg as well as from the sperms. So in that sense, he actually at that moment formulated the chromosomal theory of inheritance, that emphasizing that the chromosomes are the major site where inheritance occurs. After which, there was another scientist named as Bjorton Bjorson. Beer Bjorson was a scientist, and what he actually did was Bjorson was a scientist who Bjorson was a scientist who actually gave further upon his observation, he further emphasized the point that chromosomes in the germline cells were haploid or reduced half to the cells to the number of chromosomes which are present in the normal dividing cells. So, or the mitotic cells. So, in that sense, Pearson further emphasized that point. After which, Walter Sutton, published the work which he observed in his research paper, in his publishing paper, and named that as chromosome in heredity. This was the name of the paper which can be asked in MCQs in heredity, right? So this was the first thing that I wanted to mention. Apart from this, there was another scientist named as Thomas Hunt Morgan, who also worked on it, but we are going to discuss that at the end because he performed an experiment to prove the chromosomal theory of inheritance. So, upon such evidences which were being found out at that time, um, the Walter Sutton, collected some evidences, and then he formulated the chromosomal theory of inheritance. Number one evidence for that point was that egg and sperm contribute equal number of chromosomes to the um, diploid cell. So sperms basically have little cytoplasm because the head only fuses with the egg rather than the entire sperm fusing with the egg of the female, due to which it was then uh, known that chromosome must be present somewhere within the nucleus rather than being present in the cytoplasm, because if it would be in cytoplasm, then some of the chromosome must have been left outside the cell at the time of fertilization. So due to this, it showed that must reside within the nucleus as a result of you know e- as a result of little chromosome in sperm, and that was the point from the hypothesis that equal share of um, chromosome from sex cells would be there, equal share of chromosome from sex cells. Number two is law of segregation. What he actually what One more thing that I forget to mention about Walter Sutton's theory was that Walter Sutton, actually, when he was seeing the rapidly dividing cells of grasshopper, he actually noticed that the segregation of chromosomes during the meiosis of those cells was same as the segregation of chromosomes during Mendelian's factors, segregation of Mendelian factors during the division. So at that moment, Waterston gave that idea that since the law of segregation applies on this rule, the number of chromosomes which are present in the sex cells must be half the number of chromosomes present in the diploid cells, due to which he just gave this concept and just gave the concept of equal share of chromosome will be there. After which, number second introduced law of segregation. It verified the law of segregation that segregation of chromosome occurs according to the segregation of Mendelian factors, segregation of factors in Mendel's law. Second is law of independent assortment. It also showed that independent assortment, that the assortment of traits occurred according to the law of independent assortment of uh, mentally. Apart from which, the fourth part included the number of chromosomes being reduced to half, such that germ cells have n number of chromosomes, haploid number of chromosomes, rather than the entire number of chromosomes. It is having half the number of chromosomes in the germ cells. So these were the four evidences which were discovered at that time. But there was one question that that raised concerns regarding the chromosomal theory of inheritance. That question was that if we are having the number of chromosome, if the independent assortment is according to the assortment of factors in Mendelian trait, and the segregation of chromosomes is according to the segregation of chromosomes in Mendel's law of segregation, then why is it so that the number of assortments in traits is greater than the number of chromosomes on which those traits are present? So at that moment, in order to verify this condition, Thomas Hunt Morgan, performed an experiment. That experiment was basically performed on Drosophila melanogaster, in which what he did was he observed a fruit fly, with a mutant gene. In that mutant gene, it had the eye color white. It had Y eye color, and that white-eyed male was, having, uh, since it was male, so it was having one X chromosome, one Y chromosome, because uh, male have X and Y chromosomes, while female has X and X chromosome. Female have XX, while male have XY chromosome. And if we talk about animals, animals have, in animals, the male has ZZ chromosome and female has ZW chromosome. So these are some concepts to keep in mind. Um, what actually he did was he crossed the white-eyed male with a red-eyed female. And when he crossed the white-eyed male with a red-eyed female, what he obtained in the F1 progeny was that he obtained in F1 progeny something really shocking, which was that out of 4,252, out of 4,252 of the offsprings, 782 were were white-eyed, but 782 were white-eyed, which means 18% of them were white-eyed, and that ratio was greater than 3 ratio 1, which was obtained in Mendel's law of independent assortment. But later on, it was shocking to know that those white-eyed fruit flies were all male. So at that moment, uh, the F1 progeny was that was the case with the F1 progeny. But what actually happened in the second generation is that what he did was that he crossed a white-eyed male with a red-eyed female. When he crossed a white-eyed male with a red-eyed female, the number of when he crossed the white-eyed male with now sorry, there's a mistake. That once he crossed the white-eyed male in the second generation, what he did was that in the second generation he crossed the red-eyed male with the white-eyed female. So if we see the genotype of white-eyed male had X chromosome, which was having the X chromosome, which was having red eye color, and Y chromosome was having no gene of eye color, because in Drosophila melanogaster, the gene of eye color is a sex-linked trait. Sex-linked trait is a type of trait which is only present on the X chromosome and is absent on Y chromosome, due to which it is only present on X chromosome and thus absent on Y chromosome. Eye color, the dominant eye color was red, because it was the normal color which appears in the Drosophila melanogaster, and the mutant color or the recessive color was Y. Um, the mutant color or recessive color was W, white, because it was a mutant color, so it was a recessive one. So XW was the recessive gene, and XR was the dominant gene. Now here, XR and Y were the factors obtained from white-eyed male, and on the other side from white-eyed female we had XR, which was the red dominant, and XW, which was the red, which was a uh, white genotype, and this was the red eye genotype. And since both were, since they were female, so they had XX chromosome, due to which both of them possessed the genes for eye color. But in case of male, there were only one gene having one chromosome, which was having the gene for eye color, which was the X chromosome, not the Y chromosome. When we crossed both of them, what we actually get is that if we are supposed to cross them, right? Let's suppose if we are taking female over here, XR XW, this is XR XW, and on the other hand side we have XR and Y. Now if we are supposed to cross this generation, then we get XR in the first column, and on the other side we get XR XW, right? and on the other side we get XRY, and on the other side we get XWY. Now, if you just look into the genotype, then the ratio of genotype is, if we consider the genotype, then ratio of genotype is 1:1:1, because we have got red-eyed female, homozygous red-eyed female, homozygous red-eyed female, heterozygous red-eyed female, heterozygous red-eyed male, and heterozygous white-eyed male. So we have got homozygous red-eyed male, female, homozygous heterozygous red-eyed female, heterozygous red-eyed male, and heterozygous red-eyed, sorry, heterozygous white-eyed male. So in that sense, we have got homozygous red-eyed female, heterozygous red-eyed female, heterozygous red-eyed male, and heterozygous white-eyed male. These are the four genotypes which were present in 1:1:1, which verified Mendel's law of independent assortment in the F2 generation. And once these were verified in 1:1:1, it was also found, due to the fact that the 3:1 uh, prediction of the Mendel's law was accepted, that in this case we had three females which with red color. Sorry, we had three of the Drosophila melanogaster with red color and one melanogaster with white color, which was according to the 3:1 independent assortment given by Mendel. So in that sense, this is how the confusion regarding which confusion regarding the independent assortment was clarified with the help of Griff's Morgan's experiment. So this is all about this video. Hopefully you enjoyed it, and we'll meet in another video.